Hello, everyone. Good afternoon from PNZ to you guys. I'm so Singapore is also afternoon there. It's 1 o'clock. And wherever you're tuning in, if it is morning there, thank you. Good morning to you. If it is evening, whichever time of the day, that would be the greetings that will be going back to you. Okay. Um, Thank you all for joining in to participate in today's webinar. I would like to begin with some preliminary introduction before we move on to our actual seminar, webinar. Since 2017, the Asia Pacific Leaders Malaria Alliance, or APMEN, has proudly hosted the Asia Pacific Malaria Elimination Network, or APMEN. APMEN is a collaborative platform for 22 national malaria programs and 54 partner institutions working towards malaria eliminations in the region by 2030. APMEN plays a vital role in strengthening national malaria programs by brokering technical expertise, fostering peer-to-peer learning, and connecting countries with regional partners. The APMEN network is structured around three working groups: vector control, VIVEX, and surveillance and response. And the three interest groups, including outdoor transmission, climate and environmental change, and border malaria. Today's webinar is part of the APMEN TechTalks series, which highlights key technical topics from APMEN's working groups and interest groups, and facilitates knowledge exchange on research, implementations, and field experiences. This session is hosted by the APMEN Outdoor Transmission Interest Group, which focuses on bridging knowledge gaps in vector control, especially for outdoor transmission, and developing strategic and operational guidelines for both mainstream and innovative tools. The primary WHO recommended methods for malaria vector control are insecticide-treated bed nets, indoor residual spraying, and now also spatial emanators, all targeting indoor biting and resting mosquitoes. However, in the Asia-Pacific region, most vectors prefer biting outdoors and results in malaria parasite transmission persisting in the presence of strong vector interventions. So in this webinar, 4 leading experts will discuss their knowledge about the ecological basis of the outdoor malaria transmission problem and intervention methods or intervention options targeting outdoor biting mosquitoes. Before we begin, let me quickly go over a few housekeeping notes. So this webinar is being recorded, and the recording will be made available to all participants, and a link to access it will be shared in the chat box. So while we won't be taking live questions during the presentation, we encourage you to submit your questions in the Q&A box. We'll do our best to address as many as possible during the Q&A discussion. When submitting a question, please start with the name of the panelist you are directing it to. For example, Dr. Leo, what is the so-and-so and so? And this helps us route your questions more efficiently. Please note that we will be only monitoring the Q&A box for questions, not the chat box. That's important. You're free to chat in the chat box, but when you're posting questions, please post it into the Q&A box. We'll have 4 presentations delivered back to back, followed by a dedicated Q&A and a quick poll at the very end. Please be informed that this webinar has attracted registrations from 902 individuals from 90 countries. Based on our experience, we anticipate that more than 450 participants will join the session. Once again, thank you for tuning in, and let's now dive into our presentations. So I'm going to introduce our first speaker. That's Professor Edward Walker. Dr. Walker is professor in the Department of Entomology, as well as Department of Microbiology, Genetics, and Immunology at Michigan State University. He is a leading expert globally on the ecology and control of malaria vectors and parasite transmission. His research portfolio includes many decades of studies in many parts of the world, including Africa, South America, Philippines, and of course, PNG. He has over 300 peer-reviewed publications and have authored several book chapters throughout his careers. I am very proud to also say that Dr. Walker was my supervisor for both my master's and doctoral program at Michigan State, and it is my honor to introduce him. The title of his presentation is Timing and Location of Anopheles Biting, Brief Review of a Complex Problem, and he says in a general club approach. So, Prof. Walker, the microphone is yours.
Thank you, John, so much for this opportunity to be with you all. And I'm very glad to be with you and talk about this important topic. I would like to give a sort of a journal club approach to it, which means looking into the past, reaching back a good ways and up to the present about the emergence of this problem, and then how our research and operational control community has responded to it. Okay. I think there are three important points to consider with regard to the problem of outdoor biting and changes in the timing of biting and host seeking by Anopheles vectors of human malaria. The first is epidemiological significance. There is agreement that location and timing of vector biting are important to transmission and have implications for malaria control. Sorry, I just lost my. Here we go. It leads to the concept of residual transmission, that is, that fraction of transmission remaining after the effective control measures, sometimes related to coverage. The second important discussion point regards evolutionary significance. Now, here I'm going to take the point of view, and we can discuss this, that there is not agreement or compelling evidence that changes in timing and location of host seeking and biting in a control context are related to an evolutionary process, although it is often implied or concluded. Lastly is historical significance. Variation in location and timing of vector biting is an old topic, long considered, well anticipated, and investigated within the original IRS framework of the first malaria eradication era and even before. So this is really not something new that we're facing, say, post-ITN or LLIN era. Now, Perugini et al. In 2023 estimated residual transmission with an LLIN intervention in Burkina Faso. This creates some context for us. There were two species of vectors in that situation, and they quantified when people were awake in villages and whether they were outdoors or indoors. And they also quantified when people were asleep, meaning they were indoors. Simultaneously, they quantified the biting pressure on people using biting counts outdoors and indoors over time. And when they did this, they were able to estimate the residual transmission as a fraction of potential transmission if there had been no LLIN coverage. The blue line shows the estimated EIR, or entomological inoculation rate for unprotected exposure, and the red line shows the estimated EIR given protection indoors and human outdoor behavior. And their estimate was that the residual transmission due to vector-human contact outdoors in an early evening and early morning hours was 10% of the potential transmission. So this gives us a hard number on what we mean by residual transmission in a context like this, where there's ongoing control. Now, interestingly, it's important to point out, and they did point this out in their study, that they found no major shift in mosquito biting rhythms in response to ITN or differences between indoor and outdoor catches. Now, this is an area that's undergone long-term LLIN implementation. So in this case, and I think it makes a good background for us, there has been no shift towards early evening biting nor towards outdoor biting. The predominant biting remained indoors late at night, as you can see for the two species in question. Therefore, the estimated residual transmission shown in the previous slide would occur even if the houses were mosquito-proofed by screening entry points into them as opposed to LLIN coverage. These are species whose populations carry significant pyrethroid resistance, which could explain the lack of a change in the predominant timing and location of the host-seeking phenotype. Now let's go back to the Southwest Pacific and go back into time a little bit. A key paper, And I hope that people have read this, but if you haven't, have a good read of this. It's by Brian Taylor. It was published in 1975, titled Changes in the Feeding Behavior of a Malaria Vector, Anopheles ferrauti, Following Use of DDT as a Residual Spray in Houses in the Solomon Islands. Taylor worked on San Cristobal Island, as shown here, and he observed that before DDT IRS was rolled out, and the top two graphs on the left, the pattern of biting in time and space by Anopheles ferrauti was outdoors and indoors, approximately 50% in those two locations, and was primarily early in the evening and slowly declined until the following morning. After spray, this pattern shifted dramatically, as we can see in the lower two graphs on the left. The host-seeking timing in particular was pushed to early in the evening, and then it declined much more rapidly, so there was an increase in the concavity of this curve of timing of biting. So after DDT IRS, timing of biting of Anopheles ferrauti reshaped to predominantly early evening, but there is still indoor biting. So Taylor followed this up with an experiment. He had one unsprayed house and one sprayed house. And in the unsprayed house, he collected indoors and outdoors and looked at the distribution of biting over time, but time isn't shown here, but just location and days after spraying. And you can see approximately 50% of the biting was indoors, meaning the other percent of biting was outdoors. Now in the sprayed house, by contrast, biting indoors was greatly diminished, as shown by the black circles, and this rose over time to 160 days. DDT IRS appeared to cause a house entering deterrence. This was non-contact, non-house entering effect, he concluded, I think correctly. But the mechanism of this effect, a non-contact one, is unknown and is hard to figure given the low vapor pressure of DDT, which is a solid at these temperatures. So Taylor also tabulated his data, and this is very useful, not quite raw data, but enough that we can make some inferences beyond what he presented in his paper. And there are two things I want to point out in particular from this important key paper in this regard. First of all, it's important to note that the DDT IRS had a population reduction effect. This is something he doesn't actually highlight that much in his paper. It was about 60%. So DDT killed mosquitoes and reduced density. That's shown in green. The other important effect was this behavioral push outdoors effect, which he interpreted to be a deterrence from house entering. And it the indoor to outdoor ratio, or outdoor to indoor ratio of biting, was about 1-to-1 before spraying. And after spraying, in the red box on the right, that was reduced to about 2 outdoor bites for every 1 indoor bite. And that was a very profound effect, and as we can see, lasted for quite a long time. So there was a push to outdoor biting, and there was a change in the distribution of timing of biting. Now, it's important to reflect on how this might have happened. Another paper that's relevant to interpretation of Taylor's findings came much later. This is a very elegant study by Tanya Russell, published in 2016 with the colleagues shown here. It was a mark release recapture experiment trying to understand if Anopheles ferrauti is heterogeneous in location and timing of biting. They concluded the opposite. This species in the Solomon Islands was homogeneous. For example, they could release a female who's marked, and she might bite early outdoors one time and in a later gonotrophic cycle bite late indoors. So that was not a phenotype that was fixed in various individuals in the population, but rather there was a lot of homogeneity. So that population homogeneity suggests there is no phenotype upon which evolution might select for outdoor early biting. Very fine study if you'd like to look that up. Now back to this push effect. I just want to mention this because I'm leaning in the direction of having us think that there can be chemical modification of behavior, which does not require us to conclude that there's an evolutionary change in the population. But we don't fully understand the mechanisms by which this can happen. Back to Taylor, 1975, how does DDT act to push Anopheles outside and to earlier biting if DDT is not volatile? It's a solid at room temperature and has very low vapor pressure, as this tabulation shows. At this low vapor pressure, it should not volatilize. Now, one possibility is that there's a co-distillation effect, and this has to do with physical chemistry of DDT interacting with water vapor. When it's mixed with water in either the wettable powder or emulsifiable concentrate formulations and applied to wall surfaces as a spray, DDT could vaporize with small droplets of water as the water droplets themselves vaporize. And this is a phenomenon called co-distillation. If that happens, then it's somehow behaviorally active in the atmosphere surrounding the house. This is one possibility that was never explored but could be done. In agriculture, this is shown to happen. However, this DDT push effect is not always present. So let's be cautious in concluding that it's always going to be there. Shadrawi, much earlier, working in northern Nigeria, did a very similar study, quantified human behavior outdoors and indoors in villages in northern Nigeria, as shown in the left two panels on the top, and also quantified indoor and outdoor biting of Anopheles arabiensis at this location, all in the relationship of unsprayed or DDT IRS sprayed houses. What do we find from this study? People in these northern Nigeria villages are outdoors late, but most people are indoors at night and they arise early. Both indoor and outdoor biting, given a combination of mosquito and human behavior, is a normal part of this transmission system. It existed prior to the implementation of the IRS. Anopheles arabiensis will bite outdoors, it will bite indoors. It will bite early, it'll bite late. In fact, biting counts should have started sooner in the afternoon and lasted longer in the morning if you look at the graphs in the bottom left. In villages with houses sprayed with DDT IRS, mosquito populations were pushed lower than before spray or non-sprayed areas, comparing the left and right lower panels. But DDT appeared not to offer this repellency or deterrency remodeling of mosquito behavior under these conditions, something Taylor did show. So there was no evidence for evolutionary change of this kind, but some evidence for vector control based on density reduction. Let's go back to the Solomon Islands and examine this paper by Hugo Bugoro and others, and you'll recognize these names of these authors, with Tanya Russell in the last author position again here. Their studies were done in a different location than Taylor's, and this was done really as entomological survey analysis during the course of a rollout of two different interventions, one bed nets, Net Protect with deltamethrin, and the other indoor residual spraying using Icon 10CS, which is a lambda cyhalothrin formulation, as you can see here. The top row on the right panel shows when the entomology surveys were done, before and after the interventions. The middle row shows when bed nets were distributed, and the third row shows when the IRS was done. Now, this is a complicated set of figures, but I'd like you to just focus on B and D, please, the village of Pala. And what they observed was that before the IRS with the Icon was done, there was approximately an even outdoor and indoor distribution of bites by Anopheles ferrauti. And that's shown in panel B with the two blue arrows. As we move to panel D, after the implementation of the bed nets and with IRS, we see a separation with more outdoor biting and relatively less indoor biting, but really no change in vector density. And nor was there a change in parity rate from before to after these interventions were implemented either. So these were relatively weak interventional effects. Now, how could there be a push to outdoor biting in this case? Well, one possibility is that lambda cyhalothrin, which is well known to have contact-mediated excitoropellency and killing effects, could have resulted in contact-mediated push to outdoors of Anopheles ferrauti. It's unlikely that it could have the kind of DDT effect, which is pre-contact atmospheric effects, because it has a very, very low vapor pressure, but it could cause contact-mediated change in behavior. Accordingly, any increase in outdoor biting could be a consequence of the excitoropellency effect of the Icon CS with a lambda cyhalothrin active ingredient. And this change does not have to be attributed to an evolutionary change in population host seeking and biting behavior. It's unclear if the authors were leaning in the latter direction, but they did suggest that there was a change in biting behavior that was a result of an evolutionary change, but we don't have to accept that as the explanation. And then the last paper I wanted to refer to was that by Eddie Thompson and co-authors shown here. This was published in 2017 in the Journal of Infectious Diseases, a key paper for all of us to examine. A robust study that encompassed LLIN intervention, analysis of human behavior, vector behavior, estimates of transmission, and consequences to human malaria infection. And this was done in Papua New Guinea. In this place, there was a highly outdoor biting Anopheles fraudi species 4 as the primary, I think the sole vector that they found there. Now, they observed minor but epidemiologically important shifts to early biting time within 2 years of LLIN rollout. And this is a very subtle effect. And the two top panels on the right show this effect. In Cocofine, one of the areas where the studies were done, there was about a 1 hour earlier shift in biting time. And in the village of Mauno, there was about a 2 hour earlier shift. Now, there was really no shift from indoors to outdoors because this species is predominantly an outdoor feeder anyway, about 84% outdoors bites and about 16% indoors bites. So the focus here was on an earlier biting pressure. Now, how an indoor intervention caused a change to earlier biting time in a largely outdoor biting Anopheles species is left unexplained. Selection pressure seems like a weak explanation. I wondered about changes in host availability, for example. But over a 2-year period, one wonders if evolution could act quickly enough given this weak selection pressure for this change in biting time, which was subtle but epidemiologically significant. Okay, some observations and conclusions. First of all, and I didn't comment about this at all during my narrative, but there's a problem of inadequate experimental field design to address the question properly. This is my opinion, and it's not the fault of the investigators because they're usually doing studies in the context of a rollout of an intervention. But lacking are good controls in time in particular, where non-treated controls post-treatment are included in a before-after treatment control design. It's just a problem of the reality of the work conditions. Secondly, investigators and authors tend to use somewhat provocative language that suggests or infers evolutionary change in the phenotype of timing and location of biting or host seeking when evidence for it is lacking. This is my opinion. Rather, a chemically mediated effect modifying these behaviors through subacute toxic influences is a much more likely explanation, along with changes in host availability due to LLIN physical barriers. Thirdly, if there are chemical modifiers of indoor and late night biting pushing to outdoor earlier evening or morning biting, we need to investigate the mechanisms involved and understand these much better. For DDT, there was some kind of non-contact repellency. For pyrethroids, whether in IRS or in LLINs, this is likely a contact-mediated push. Maybe this push can be managed to avoid it. Nonetheless, timing and location of biting are clearly important, leaving epidemiologically significant fractions of residual transmission and reducing effects on some of the targeted vector species populations. Thank you very much.
Well, thank you, Dr. Walker, for a wonderful, clear, and thought-provoking presentation on the topic of timing and location of host seeking by malaria vectors, which encompasses the problem of outdoor malaria transmission, okay, which is the topic for this webinar. I'm sure some members of the audience will start thinking about the two competing hypotheses you have shed light on. One in which the change in vector biting behavior in response to insecticides as an evolutionary outcome, and the other being a chemically mediated outcome. So that to the audience, if you have any questions for Dr. Walker, you can post them on the Q&A box, okay? Avoid the chat box. Make sure to indicate the name of the speaker your question is directed to. Let us now invite our second speaker, Professor Leo Brack. Professor Brack spent most of his earlier life working on malaria and wildlife parasitology in Africa before moving to Asia. He is a medical entomologist and provides support for vector control for malaria consortium offices in Asia and Africa, specifically relating to malaria and arboviruses. Leo is also the co-chair of the APMEN Capacity Strengthening Intervention around vector surveillance in the 22 member states. He has over 100 peer-reviewed publications and 7 books to his credit. Professor Brack will be speaking to us about intervention options against biting vectors. Ladies and gentlemen, please welcome Professor Brack.
So thank you for that introduction, John, and thank you, Ned, for a great presentation. and a very warm welcome to our esteemed audience from all over our good planet. Very welcome. It's good to see you all. Hope you're enjoying the presentations thus far. So I'm going to try and focus on outdoor biting, but before doing that, I'd like to ask that we just remind ourselves that in Africa, where about 95% of the malaria burden globally takes place in Africa, um, malaria is transmitted by a relatively small number of vectors, which are overwhelmingly indoor biting, indoor resting, and strongly anthropophagic, strongly attracted to humans, indoor biting, indoor feeding. In Asia, the flip side is true, that there's a relatively large number of vector species involved in malaria transmission. They belong to a fairly substantial number of species complexes, which makes them difficult to identify. It's not simple for field staff to identify exactly which species involved. But equally relevant is that overwhelmingly the majority of these vector species in Asia Pacific have a preference for biting outdoors, for resting outdoors, and to complicate it even further, many of them are to a greater or lesser degree also zoophilic, so they'll feed as readily on cattle and other domestic animals as indoors. So also, again, just as context, a reminder to us that vector control, by and large, is the primary tool for malaria control globally. If you look at the proportion of the budget that gets set aside for malaria control, the bulk of the money available for malaria control goes towards vector control. So here is an interesting excerpt from a publication by Bart et al. Here at the bottom of the screen, published in Nature in 2015. They don't come much more reputable than Nature magazine. Excuse me, a very impressive study, very large data set, which showed that between 2000 and 2015, we estimate that vector control interventions have averted 663 million clinical cases in those 15 or 16 years. Insecticide-treated nets, the most widespread intervention, were by far the largest contributor. In fact, about 68% of cases averted were due to ITNs, while indoor residual spraying contributed a further 10%. So between ITNs and IRS, about 78% of averted malaria cases were achieved just by way of those two vector control tools. Artemisinin combination therapy, parasite control contributed a further 22% towards malaria reduction. So if vector control is so important, who does the global malaria community look to for guidance as to what the vector control tools should be? And without doubt, that's WHO, World Health Organization, is the primary indicator of endorsement for specific vector control tools, approaches, and practices, and so on. So it's important to note also that WHO, when they make these recommendations, they don't necessarily look at what interventions are most effective for personal protection. WHO looks at tools that have an impact on malaria reduction at population level, community level. What are the tools that achieve a reduction in malaria at scale? So, a topical repellent that helps Joe Blogg sitting outside, WHO acknowledges that that may be effective, but it doesn't really translate into population level malaria reduction. So when you see WHO recommendations for particular tools, keep in mind that WHO has this particular perspective on what they mean by what tool is effective. They look at it from a population level malaria reduction impact. So in the context of vector control, WHO only endorses two tools. That is insecticide-treated nets, and specifically those that are pre-qualified for use, not just any old bed net, very specific ones. And then also indoor residual spraying, IRS. Again, very specific products that they will endorse. Those are the only two vector control tools that are fully recommended, endorsed, and supported by WHO. WHO also provides conditional recommendation for two other approaches as a supplementary method of vector control. And that is the use of spatial emanators. And that was only published just over a month ago, 13th of August, 2025. These recommendations for spatial emanators came out. And the other conditional recommendation is for larviciding. Take note, not larval source management, specifically larviciding. So WHO endorses ITNs and IRS unequivocally for vector control, and then conditional endorsement under very specific conditions for spatial emanators and for larval source management. And you'll note for three of them, and I'll get to spatial emanators in a moment, for three of these four, they directly address only indoor biting conditions. So we are still stuck with what to do with outdoor biting. So with regard to spatial emanators, WHO says there's conditional recommendation with moderate certainty of evidence supporting that conditional recommendation. But if you look at the bottom yellow highlighted qualifications here, they say that spatial emanators are only recommended for indoor use, and then only two specific products, and that is mosquito shield and Guardian, which are both manufactured by SC Johnson, and they're both based on transfluthrin. Mosquito shield lasts for about a month, Guardian lasts for about a year. But in addition to only indoor application, they recommend and they only endorse it for use in combination with either an ITN or IRS. So spatial emanators are not recommended by WHO as a standalone product. So just bear that in mind. So our talk today is actually supposed to target outdoor biting, and I'm going to get to that. And I'm going to talk about a whole bunch of approaches and tools, and I have very limited time. I'm going to overstay my welcome by talking beyond my allotted time, but I'll try and talk fast and try and get through these as quickly as possible. We'll go through all of these one by one. So sticking with the subject of repellents, such as spatial emanators, some people call them spatial repellents. Repellent impregnated clothing on the left here. WHO gives conditional recommendation against. In other words, WHO does not recommend or endorse repellent impregnated clothing. But they do say that at the bottom here, yellow highlighted, keep losing my cursor. Insecticide-treated clothing may be beneficial as interventions to provide personal protection against malaria in specific population groups. And clearly, those specific population groups would be for people that are not protected by bed nets or IRS, and you think specifically of outdoor conditions or humanitarian situations, displaced people, people that find themselves in a situation where they don't have access to ITNs or IRS or other forms of protection. Treated blankets and top sheets here towards the middle of the slide. This is not dealt with by WHO, and that's simply because there are very, very few studies that have been done on blankets and so on. It's mostly in Afghanistan and so on. Very few of those studies, very little evidence available, so there's very little to judge the merit of treated blankets and top sheets. And you may wonder why I talk about blankets and top sheets, and that's because, for example, in Africa, you will frequently find people sleeping outdoors because it's simply too hot to sleep indoors. So people do use these treated blankets and sheets potentially for outdoor protection. Topical repellents, again, WHO gives conditional recommendation against topical repellents because of the low level of evidence supporting population level impact on malaria reduction. So it is not recommended for malaria control at the community level. Spatial emanators or spatial repellents, we've already dealt with that, so I'm going to move on. The other one, Wulan, this extra panel keeps obscuring what I can see on the slide. But anyway, refers to attractive targeted sugar baits. This is an approach that's been receiving a lot of attention in recent years. A lot of research has been focused on that. And that is based on the fact that although female mosquitoes seek blood, they need a blood meal for egg maturation, they need blood for the eggs to mature properly. Both male and female mosquitoes also need access to sugar-rich fluids for energy supply. And so if you take a sweet substance, fruit juice, something like that, and you incorporate a toxic substance into the sweet fluid, ideally also combine it with an aromatic compound that acts as an attractant for the mosquitoes, and you place that in a membrane-covered package, and you hang it up on the wall. This attracts both males and female mosquitoes. They come and pierce it, take the meal, and they die. So as I said, a lot of research has gone into that in recent years. This is how you apply it. You hang it on the outside or inside the home. But so far, there's been very little evidence to support large-scale use of this, primarily because of the loss of physical integrity of the particular product. It suffers a lot from damage by rats and other physical abrasion. And also, there's inadequate evidence thus far that it is effective for malaria reduction. The work that's been done has been only in Africa. I'm not aware of any work that's been done in Asia. So there's still a lot of research that needs to be done. And yeah, okay. One approach that I particularly like very much, and I wish it would receive more uptake and attention, is insecticides. Insecticides, from the name, you can tell it's chemical compounds that affect endoparasites and also ectoparasites. Ivermectin is one of the products that functions as an insecticide. Ivermectin, here highlighted in yellow. It's been used for decades in the fight against things like river blindness, onchocerciasis, also used for lymphatic filariasis. but millions upon millions of doses have been given to humans through mass drug administration to combat river blindness very successfully. Extreme left photo shows you one of those helminths, a nematode that causes river blindness. It's been used very effectively. River blindness has been dramatically reduced. Ivermectin has also been used across the world in probably billions of cases for application for worm control in cattle specifically, very effective against internal helminth parasites. So in the process of administering ivermectin, both for human application and also cattle, it became evident that mosquitoes feeding on a recently dosed human or animal die. And they will continue, mosquitoes feeding on a treated human or cow or so on will die for 3 weeks. And if you use a long-lasting formulation in cattle, for example, that effect will last for months. So if you're in a situation such as in large areas of Asia, India, Greater Mekong sub-region, etc., where many households are associated with several cattle, excuse me, in attendance. So cattle owners would welcome administration of ivermectin because it helps improve the condition of the cattle by reducing the worm load, and it will kill off each and every outdoor zoophilic mosquito that feeds on it for X number of weeks. So, I think this would be a perfect way to address residual malaria in remote population situations where country is approaching elimination but struggling to achieve dealing with residual malaria. Anyway, this has not received endorsement from WHO because the evidence is still incoming, still not adequate evidence for WHO to make a final recommendation. Outdoor spraying, we're talking about outdoor biting mosquitoes. If a mosquito feeds outdoors, it has to rest somewhere. If you've got a full belly of blood, you'll try and find the closest site to rest, and under the eaves or overhangs of dwellings is an ideal protected area. So if you spray there or on vertical walls around cattle sheds, etc., also ideal opportunity. But once again, there's inadequate evidence that this has the potential for malaria reduction, and therefore WHO has not come up with an opinion regarding outdoor spraying. Fogging or space spraying, again, WHO gives a recommendation, conditional recommendation against fogging or space spraying, not for. And it says space spraying is not recommended. It's used mainly for arbovirus vector control during outbreaks of dengue and so on, to achieve very rapid response in the vector population in those outbreak areas. But, it's a fairly expensive, labor-intensive intervention. So it's not being applied in malaria control extensively, and the evidence suggests that this is not a very effective way for cost-effective malaria reduction, given the challenges that face malaria budgets and so on. Mass trapping, this would refer to where you deploy en masse large numbers of CDC light traps or other trapping methods to reduce the population of outdoor biting mosquitoes. Again, WHO doesn't say anything about this as a recommended method. Almost certainly because this is very expensive, very labor intensive, and so it's just not a very practical way to go about reducing malaria at scale. So yes, it works in a very local situation, but it's going to cost you a lot of money, and you need to apply it constantly with a good team of people to deploy it. So WHO is silent on the use of mass trapping. This one, larval source management. WHO does not recommend larval source management, but it does recommend conditional use of larviciding, particularly where aquatic habitats are few, fixed, and findable. In other words, they do not recommend it for large bodies of water, dams, marshes, um, rivers, streams, and so on, but rather in situations like Anopheles dirus or some other species that breed in small, confined, localized habitats where it's easy to spot treat those breeding sites. Problem is that you need to be able to find them and then treat them. So larval habitat modification in the broader context, larval source management, is no recommendation can be made because the evidence was deemed to be insufficient. So larviciding, yes, conditionally, where the breeding sites are few, fixed, and findable. Problem is you've got to find your own money. WHO or Global Fund is not going to provide you with money to do this. It has to be domestic domestic funding. Larviciding is becoming more appealing, and there are more and more publications appearing that are encouraging the use of larviciding because it can be applied locally by community members in a self-help way for local reduction of mosquito populations. But at programmatic level, countries like Pakistan are combining larviciding with more recent technology innovations like the use of drone technology, using drones to locate these few fixed and findable breeding sites, and then also using drones to actually take the larvicide and delivering it to difficult to access breeding sites. Like very often, especially in Africa, your riverbeds that recede in the dry season to small pools where a lot of Anopheles arabiensis and Gambi and so on breed. They're surrounded by dense beds of reeds and so on, difficult to access. So those are ideal situations where you can then use a drone to go and deliver the larvicide. So again, excuse me, WHO only recommends larviciding as a supplemental method, not larval source management in general. Sterile male technique, sterile insect technique, is based on the principle that you rear millions of mosquito larvae of the target species involved. You have to target a specific species. You rear millions of larvae. When they reach the pupal stage, you sieve out the male pupae. The male pupae are smaller in size than the female pupae, so you can separate the two sexes. You subject the male pupae to radiation, and then it renders the male adults infertile, sterile, and therefore when they mate, you release them by the millions into the environment, and when they mate with wild fertile females, it results in infertile, sterile offspring. Problem is, you need to rear these in their millions, and you need to release them continually, regularly, to achieve a sustained impact. So it becomes expensive, labor intensive, costly, and although it's being used very effectively against a number of different species, in a veterinary context, screwworm, in Central America, Libya, and so on, to eradicate screwworm from entire countries or regions. Tsetse fly also very effectively used against tsetse fly to cause local extinction of tsetse flies. And then a number of different agricultural pests have also been targeted very successfully. So again, the method works. It's just impractical, not cost effective for malaria control. Gene drive technology is receiving a lot of attention, very enthusiastically, but it's still in the early development stage. This method, there are two approaches. You can either try and achieve population suppression or population replacement. For suppression, if you look at the bottom left here, the graph there shows that you try and suppress the population, ideally to extinction, which is going local extinction, by way of identifying a gene in the target species that you want to target. You find a gene that causes sterility in the offspring. Then you need to find a second gene that and that gene that causes sterility that's the effector gene. Then you need to find a drive gene that you combine with the effector gene that will drive the effector gene in a greater than 50% inheritance ratio. Remember, if if a male and a female mosquito copulate, their offspring will inherit a particular trait in a 50-50% ratio. So that's not good enough for the purposes of what you're trying to achieve. So you need a gene that will force that desirable gene into a near 100% of the offspring. So there are two jobs to perform. That's population suppression. Population replacement is where you identify a gene that renders the female refractory to infection. In other words, the ability of the female to be infected by the pathogen is reduced. So same principle as with the suppression. You need the effector gene and you need the drive gene combined to place into female offspring, release them, and then they will achieve this desired effect. It's still in the experimental phase. There are multiple studies, or several studies. Target Malaria being one institution doing excellent work. In Africa, I'm not aware of any of this work going on in Asia, but in Africa, there are very strong concerted efforts in combination with European universities working on this. So it's not a method that's currently anywhere near large-scale deployment stage.
So we are
But
running out of time, if you could.
okay, sorry, I'll hurry. So this one I put in tongue-in-cheek, but it's something that really works. Several studies in Africa have proven it. Derek Shalwood in Papua New Guinea showed that it also works in malaria vector species in Asia as well. So if you stand or sit outdoors in Africa or Papua New Guinea, 96% of bites will occur at ankles and feet. And to show that it's got nothing to do really with ankles and feet, if you take that same person and you strip them down to underpants and you let them lie down, those vectors will bite anywhere on the body, which shows it's a height above ground thing. So if you can convince people to sit on raised platforms, a dramatic decline in the number of bites will result. It's not going to work. You're not going to persuade people to change their behavior, to suddenly start building platforms and sit on that, but it does work, does definitely work. What's this one? Zooprophylaxis is the principle where if people keep cattle in proximity to where the humans are, the cattle will draw zoophilic mosquitoes away from the humans for the mosquitoes to preferentially bite on the cattle. The opposite also applies, that those cattle will then could potentially attract more mosquitoes to where the humans are. The evidence is mixed. It's not clear that zooprophylaxis actually works. So we end up with, what can we therefore do? Bottom line is we do not have a silver bullet a tool to address outdoor biting. The best we can do is to engage in public campaigns to inform the risks to the public, explain to them that topical repellents will work, try and reduce breeding sites as best you can, and do this in an integrated vector management approach. Sorry, I'm out of time, but just please remember that LLINs and IRS is part of integrated vector management for outdoor biting mosquitoes as well. There is no such thing as a malaria vector that only exclusively bites outdoors. A good percentage of those outdoor biting mosquitoes will come indoors periodically and be exposed to either the impregnated bed nets or IRS. And so you get an erosion of that outdoor population taking place because of indoor methods. So I'm sorry I overstayed my welcome, but thank you for your attention. Sorry about that, John.
Okay, thank you, Professor Brack, for a very important presentation. I'm sure many in the audience have enriched their knowledge about the various vector control methods that was discussed in the talk. So let us now jump straight to our third speaker, Miss Carusele, Michelle Carusele. She holds a master's in vector biology from Liverpool School of Tropical Medicine. She is currently a senior scientific officer with PNG Institute of Medical Research and a doctoral student in the College of Medicine at James Cook University. Ladies and gentlemen, please welcome Michelle. I think, Ulan, are you going to play the pre-recorded? Yes, please.
Can you hear the audio?
Yeah, I can hear you, yes.
In the audio from the PowerPoint?
No, I can't hear it.
Oh, wait a sec. Wait a moment. I'm trying to fix this. Okay, good. Thank you, Dr. Paul. But we can't hear the audio.
Dr. John, could you please go to there?
We conducted it.
It's playing now. I could hear.
If we cannot hear the sound, could you please go to the fourth speaker? The
I thought I had.
capacity.
Okay, well, let's do that
looking
then.
at the impact of Guardian,
Yeah,
a
go ahead.
transfluorine spatial emanator, against Anopheles punctulatus group. Briefly, spatial emanators are devices that disperses volatile active ingredients or insecticides into the air, and that creates a safe, protected space for people from mosquito bites. So the picture here shows a typical coastal village setting in PNG, where you have the main house and these common sheltered outdoor paradomestic structures. So these emanators have insecticides with repellent properties. So they obviously repel mosquitoes, but they also affect the mosquitoes' host-seeking behavior, either by masking the host from the mosquitoes or inhibiting mosquito biting or blood feeding. And they can be used indoors or in outdoor spaces. There are two types of spatial emanators. You have active spatial emanators that requires an activating mechanism, for example, the mosquito coil. And there's passive spatial emanators that doesn't require an activating mechanism, but relies on the ambient local temperature and airflow to emanate the insecticides. So the Guardian is a passive spatial emanator. So why use passive spatial emanators? First of all emanators have the potential to provide protection from mosquito bites, especially outside of the conventional indoor vector control tools like insecticide-treated nets, ITNs, or IRS. The good thing about passive spatial emanators is user compliance is minimized. So you open a packet, hang the device or emanator, and it starts emanating the insecticides or continuously emanates, providing around-the-clock protection. And they can be complementary tools to use with ITNs or IRS. So there's been a lot of interest in generating evidence over the last decade or so for spatial emanator, especially as a malaria control tool. So here are some of the recent studies that are showing promising evidence of protection against Anopheles, in particular high indoor protective efficacy against this malaria vector species. There's also evidence as well, some evidence showing reduction in malaria incidences. And furthermore, there's now a growing interest in looking at its impact against outdoor malaria vectors, especially for outdoor protection. Now, in August of this year, the WHO recommended spatial emanators for malaria vector control and pre-qualified two of the first products, including the Guardian spatial emanator. However, this recommendation is based on indoor efficacy or for indoor use only. Therefore, our rationale for the study is, can spatial emanators provide outdoor protection? So in Papua New Guinea, malaria is a major health problem with 1.5 million cases reported in 2023. And the only intervention tool we use in our national malaria control program are ITNs, or insecticide-treated nets, that is rolled out nationwide. And malaria is transmitted by members of the Anopheles punctulatus group. However, these vector species are outdoor biting species. The NAPMAP project, the study aims to evaluate the efficacy and acceptability of the Guardian to reduce mosquito lands indoors, as well as, most importantly, in outdoor paradomestic spaces in PNG. Now, we conducted a study in 2 villages in Madang, where 36 houses were randomly selected into 3 treatment arms. A control arm, the second arm with emanators only inside houses, and the third arm with emanators placed inside houses, as well as their associated sheltered outdoor paradomestic structures. The primary outcome measure was protective efficacy, indoor and outdoor, measured as landing inhibition. and we measured that using the human landing catch. We also used the human landing catch to determine a secondary outcome measure, which are landing weights indoor and outdoor. We also used an infill sentinel cage bioassay to monitor emanation of the devices throughout the study. And we did that by exposing laboratory Anopheles variety mosquitoes to these devices in the field. And we looked at 60 minutes and 24 hours post-exposure knockdown rate and mortality, respectively, as well as the probing rate 60 minutes and 24 hours after exposure. So onto the results. Here we're looking at the indoor efficacy. Here we show for landing rates, the median Anopheles collected or landed per percent per hour over days since deploying the spatial emanators. So our IS landing median lands inside houses was 3 lands per percent per hour. And there's a lot of variation in our landing rates. However, using the landing rates, we determine our protective efficacy using a negative binomial regression model. And that is calculated as 1 minus IRR times 100. So bringing your attention to the protective efficacy graph, you could see that the duration of time or the time points shaded in green shows the period of time where there was significant high protection against all Anopheles inside houses. The pie chart below simply shows the vector composition since deployment. We predominantly observe Anopheles priority towards the end of the duration of the study. We saw that Anopheles colensis was more predominant. So indoor efficacy was about 140 days or approximately 5 months. In terms of outdoor efficacy, the highest median landing rate was about 15 lands per person per hour. And protective efficacy was, again, high protection lasting up to 140 days against Anopheles in outdoor spaces. Here in our study, we show especially the indoor and outdoor arm was the better controlled strategy. And our efficacy here may be species specific or impacted by the vector species seasonality, that is the abundance per time point, which we plan to analyze further in the near future. Now, looking at the residual insecticidal activity indoors, we observe high knockdown rate, which is more than 50% knockdown, lasting up to 337 days. Furthermore, we also observed a low probing rate, 24 hours post-exposure, also lasting up to that time point. Further, in outdoor spaces, residual insecticidal activity, high knockdown rate lasted only up to 171 days. And this is the same for low probing rate as well. So we saw low probing rate up to 171 days. And after that, we observed decreased residual insecticidal activity in these outdoor spaces. To conclude, this is the first time we show that the transflutrine emanator provides outdoor protection in PNG against the local malaria vectors over an extended period of time. And using the emanator inside the houses or in these outdoor sheltered peridomestic spaces was effective at reducing more than 50% human vector contact over 140 days or approximately 5 months. We also indicated that there was a show presence of transflutrine indoors lasting up to 392 days. However, this was negligible by the time point in outdoor spaces. So building on this work, we plan to continue with experimental heart trials as well as a field evaluation on the impact of SE, looking at malaria epidemiological outcomes. And generating this evidence and the future generation of evidence of spatial emanator in the country would provide the insight needed to the National Malaria Control Program in terms of implementation of spatial emanator as a malaria vector control tool in the country. Lastly, a big thank you to all our staff at PNG IMR, as well as our collaborators and funders, and most importantly, our study participants and communities. Thank you.
Thank you, Michelle, for presenting the preliminary results of your spatial emanator study. It was a wonderful presentation, and I believe everyone in the audience enjoyed your presentations and appreciate the importance of your findings and what it means to the problem of outdoor transmission, specifically, but also transmission in general. So let us now invite our final speaker, Ms. Rebecca Vinnit. Ms. Vinnit holds a master's in entomology from Michigan State University and currently serves as a senior scientific officer at PNG Institute of Medical Research. She is currently a PhD candidate in the College of Medicine at James Cook University, and her doctoral research focuses on evaluating the effectiveness of residual spraying as a complementary intervention to existing vector control tools in PNG. So she will be presenting to us on the topic of the implications of outdoor malaria transmission in PNG. Ladies and gentlemen, please welcome Ms. Vinnit.
Thank you, John. Can you see my slide?
You have to go to the full screen.
Yeah. And now?
Not yet.
Can you swap to the presenter
swap?
mode? Yeah.
Yeah.
Hang on.
Go to the presenter mode.
Check. Sorry, guys. What is this? Ugh. Share. Okay.
So go back again to the full slide. So.
Oh, yeah. Gotcha. And now?
Okay, very good. Proceed.
Thank you, John. Thank you, everyone. Good morning and good afternoon to all. Yep. So the presentation is on the implications of outdoor malaria transmission in Papua New Guinea. Malaria remains a major burden in Papua New Guinea. Previous studies show that IRS and insecticide-treated nets can effectively reduce malaria transmission intensity. Now, outdoor malaria transmission undermines these current control tools. On your right, you can see a map by Sinka and others that present a colorful distribution of major vectors around the world of malaria. And just to take into consideration that using these vector control tools, even though the color shows on the maps, one must also understand that you cannot use it in a standardized manner. So a sound understanding of vector ecology is an area that can help to guide and optimize vector control interventions. The major vectors in PNG falls under the punctulatus group, and here at the bottom is a phylogenetic tree from BB and others that shows the three major vectors: Anopheles ferrauti that comes under the ferrautic clade, and Coliensis and punctulatus under the punctulatus clade. They can be distinctively identified morphologically and also can be confirmed using molecular assays. Now, these vectors have a heterogeneous distribution. On the bottom right is the Anopheles ferrauti that is very predominant in the coastal areas in PNG, and punctulatus and Coliensis are also along the coastline, but mostly inland. So taking that into consideration, the evidence of punctulatus group outdoor biting behavior, we did 2 cross-sectional studies. One was in Central Province, and you can see that the composition in the pie chart there shows Anopheles variety being more predominant compared to Coliensis and punctulatus. The bar graph you see is the total number of Anopheline mosquitoes collected using the human landing catch. And there is a significant difference from the outdoor collection compared to the indoor collections. Now, in a more inland village setting that we did in East Sepik Province in Maprik District, the most predominant vector is the Anopheles coliensis and punctulatus. That behavior, even though they're different, they still show that there is high outdoor biting compared to indoor. Medang Province, we did a more longitudinal study, and it also tells the same story. Here on your right, you can see our hourly biting rates of the Anopheline mosquitoes. We did a collection from coastal villages, separate from inland villages, still taking into consideration this heterogeneous distribution of our major vectors. Now you can see that in the coastal village, there is this behavioral biting that happens earlier in the evening, and that is not the same for the inland villages where it happens later on during the day, during the night, later at night. But despite this difference biting period, overall, the outdoor biting behavior, irrespective of different heterogeneous distribution or the biting behavior, the outdoor biting mean biting rate is higher than indoors. So these are some evidence of the ITN's impact on malaria vectors and malaria in PNG. This is a study, the graph on your left, from Rayma and others, where when ITNs was initially rolled out in PNG in 2008, they did a trial of before and after in the same villages where it's coastal and inland, taking into that consideration. And you can see that before the intervention indicated by the arrow on the graph, there is high mean biting rates, and afterwards it was reduced. Now, ITNs with other malaria control programs all contributed in reducing malaria incidence in PNG, as you can see in the graph on your right, from 2008 to 2014. Now, Rayma and others also raised concerns about outdoor malaria transmission. And with this indicator on the graph on your right, it shows that there is an upsurge in malaria in PNG in 2015. So with that, there needs to be another complementary vector control tool that needs to be with ITNs to address that limitation. So my study aim was to evaluate the role of outdoor malaria transmission in sustaining malaria in PNG and to discuss its implications for the effectiveness of current vector control interventions. Currently, ITN is the only vector control that's been rolled out in PNG nationwide. So the NutNut project came up with 3 complementary vector control tools to use. As you have seen with my previous the presenter, my colleague, Michelle Catusella, presented on spatial animators. For me, I am looking into residual spraying. We knocked out the term indoor residual spraying because we were not only spraying the inside surfaces of the house, we were also spraying the outside surfaces of the household. And that's to take into consideration that outdoor biting and targeting that population of the mosquito. So the study sites were taken from the North Coast region of Medang Province in PNG, 4 villages. The triangle shapes are our coastal villages and the circles are our inland. The red colors are the treated villages and the green are our control villages. So we did an interrupted time series study design where there was 12 months intensive entomological methods that we use monthly, and after the intervention, 12 months. And the residual spraying product that was used was fluorofusion. That's a combination of clotinidine and deltamethrin. For the entomological survey, we did 3 methods. That's human landing catch, barrier screens, and indoor resting collections. But for this presentation, I will only show you the preliminary results from the human landing catches. So for the human landing catch, it is a 12-hour collection. Starts 6 o'clock in the afternoon and carries on through the morning the next day. We sampled mosquitoes indoors separate from the outdoor collections. In each of the sites, there are 8 houses that we try as much as possible to maintain them throughout this survey. The mosquitoes that were sampled were brought back to the lab, and all the anaphelines are pooled, and they are dissected and analyzed to detect Plasmodium species to determine the entomological inoculation rates. So here's our preliminary results. The anaphelines mean biting rates. You can see on the left end, the indoor biting rates per person per night is the same as the outdoor biting. The red boxes are our control villages, and the purple boxes are our treated villages before and after. It is seen that the early results indicate about a 3-fold reduction in indoor biting rates and a roughly 30% reduction for outdoors. The far end graph is the duration of the efficacy of the product being used. So they did a 24-hour mortality wall cone bioassay for the treated houses for 10 months after the spraying. And you can see that there's low deterrence indoor compared to outdoor. So these preliminary results suggest RS may have a stronger effect indoors with more moderate impact on outdoor transmission. Here is our preliminary results from the lab that shows the minimum infection rates of the Plasmodium detection. So using the indoor human landing catch samples, this graph shows the infection rates of our pooled anaphelines, treated village from control village, the before and after, and also for the outdoor. And this graphs and table shows the impact of RS on minimum infection rates in both indoor and outdoor settings. Treated villages were reduced by approximately 2.4-fold significantly, indicating a strong effect of the intervention indoors, and outdoor minimum infection rates were moderately reduced by about 1.6-fold. The control villages showed only minimum reductions. So to conclude, outdoor malaria transmission limits effectiveness of ITNs and IRS. P&G major vectors all exhibit outdoor biting behavior despite of their heterogeneous distribution. Now, P&G ITNs roll out in 2008 did reduce biting rates that also contributed with the other malaria control programs, reduced malaria incidence from the period from 2008 to 2013, but there needs to be a further complementary vector control tool. So our preliminary findings show that RS strongly reduces indoor biting with a moderate effect outdoors. However, outdoor biting Anaphelis punctulatus continue to feed outside. This highlights these gaps of protection, and these results are based only on 80% of analyzed samples that we're still working on. So trends may be refined as full data set is completed. So this suggests that complementary strategies targeting outdoor and early evening biting may be needed to further reduce transmission. I would like to acknowledge my supervisors for this research, PhD advisors, collaborators and partners who have helped us during the intervention, and also the field and laboratory team of PNG-IMR, our funders, and also our participant. Emtasol na thank you to.
Okay, thank you. Thank you, Rebecca. I really enjoy your presentation, and I'm sure the members of the audience did as well. We really appreciate you sharing your preliminary results and what it means for the problem of outdoor transmission in PNG and probably elsewhere. Again, to the audience, please post any questions you have to Rebecca or any of the previous speaker in the Q&A box, and be sure to indicate who the question is directed to. So that is the end of our seminars presentations for the four speakers. We shall now jump to the Q&A session. So I have with me 4 questions, one for each speaker, and we will go through them in the order of the presentations. That is beginning with Professor Walker and then finishing with Ms. Vinit. After each speaker has responded to his or her prepared question, there's also questions from the audience. I will take one question from the audience for each speaker, and then sort of ask you about. Right. So I understand that Prof. Walker will be exiting us in probably 5 minutes. I'm going to ask you this question now. So Ned, thank you very much for a thought-provoking talk in which you pointed out that there is no compelling evidence supporting natural selection as the cause of change in the dynamics of timing and location of host seeking for mosquitoes in response to the insecticide-based vector intervention, right? Yet, this notion appears to be prevalent among malaria vector biologists, with numerous research articles attributing these changes to the evolutionary process as opposed to the chemical effects. Why do you think that is the case? And what could we do to change this sort of perception?
Why do I think it's the case that people think that way, or why do I think it's the case that the evolution doesn't seem to be happening?
No, why do you think that many people, there's a, even though with very limited evidence to support that hypothesis, there is still a good number of proponents of it, and there's a bunch of articles being written about it.
There are, and I need to point out that I was expressing my opinion, but I think it's an informed one. There are many pathways the vector populations could take in evolutionary response to insecticide-based interventions. The most common one is the evolution of insecticide resistance. And there, the evolution is real. People have quantified changes in gene frequencies. Those are related to resistance properties such as knockdown resistance or metabolic resistance, and so on. So we know that evolution operates on vector populations. However, when we start to think about behavior, these are things that are, first of all, somewhat difficult to measure, but we have very, very little information on the genetic background that contributes to behaviors like indoor or outdoor biting. The same with host selection. So we might expect, in the context of, say, outdoor biters, the selection pressure might be pretty minor to change behaviors. You think the selection pressure might operate strongly first on resistance factors. So if that's not the case, then selection pressure on behavior modification ought to be minor as well. I was more trying to be thought-provoking with the audience here than drawing strong conclusions.
Okay.
But the other thing is maybe shifts in host selection as well. If human beings are covered by insecticide fabric material, well, just bite a pig or a dog and avoid it entirely. So those are my points of view, and I hope I kind of addressed this, but I think the rush to concluding there's evolutionary change that concerns me because it influences the thought processes of funders and program managers. And that has implications for the way we go about doing things.
Good point. Thank you, Professor Walker. There is another question from the audience that I would like to, pose to you. Then the question goes like this: do outdoor biting species become indoor biting when malaria elimination program during a malaria elimination program? I think this is a question about understanding how these dynamics and like what are the indoor biters and outdoor biters? Is there any real difference between them? Are they just So it follows on from this hypothesis or chemical effects. So do outdoor biting species become indoor biting when malaria elimination during a malaria elimination program? Sounds simple, but I think it's interesting.
Not sure I've ever heard of that.
Yeah.
A change from outdoor to indoor biting? I don't know. I guess I'll throw that out for others to think about. It is a behavioral plasticity phenomenon. We see that a lot of species are quite plastic, and maybe the perception of what is indoor and outdoor to a human being is not the same as a perception to a mosquito. It may be more where hosts are and where host factors are emanating from that's related to aggregation indoors or outdoors. So we may need to change our thinking there. Otherwise, I really am not quite sure how to answer that question.
Okay. Well, okay. Thank you very much, Prof. You may exit if you feel like you're ready to go. But thank you very much for your participation.
you all.
We really
Bye-bye.
appreciate it. Yeah.
Thank you.
Okay.
Thank you, Ned. Thank you for your time.
Okay, so our next question goes to Professor Brack. So thank you very much for a wonderful seminar on the various vector control interventions that humanity has in its vector control toolbox. Among them, some are traditional methods like the IRS, ITNs that are deployed in large scale and in and others such as fogging and larval source management are deployed in specific circumstances, so small scale intervention. And some are new or innovative methods such as gene drive and indoor spatial eliminators like you have just discussed. So with regard to the new methods, now Michelle and others investigators have shown that spatial eliminators work. And WHO has recommended 2 of these products for malaria vector. Okay. Now, what is your opinion about the sustainability of the spatial eliminators as a vector intervention method, especially in rural endemic areas? Okay. And does it have the potential to be deployed in large-scale intervention program?
Thank you, John. I mean, that recommendation from WHO came very recently. Hardly a month has gone by since that recommendation. And then, as you know, as we explained, very specifically for indoors, for only 2 products and only in association with ITNs or IRS. And that is because what the evidence has shown, the experiments that have been conducted in recent years, that's what the evidence clearly shows where spatial emanators are very effective. They may very well be effective under different conditions as well. I certainly hope so. And Michelle is doing excellent work to contribute to that evidence. Sustainability, there's adequate evidence that, it's an effective method for indoor application. And I think also we mustn't forget there are certain situations where it will really find added value, and that is under specific conditions like humanitarian emergencies where, there's mass migration of people, they get housed in tents and so on.
Okay.
It's difficult to hang up an ITN in a tent or to apply indoor residual spraying. And that is where the real value of spatial emanators will come in. And that is also where international agencies may be willing to provide funding, because at the end of the day, it becomes sustainable if there's money available. We all know that there's a serious shortfall in funding for malaria control programs globally. What available money is available goes towards, by and large, provision of bed nets. So there's very little money left for Global Fund or anyone else, and they're very unlikely to provide money for purchasing at scale things like spatial emanators. That will, for the immediate future, will have to come from domestic sources or philanthropic sources and so on. So, or, end users themselves. So unfortunately, that's the reality, that while spatial emanators may have proven themselves very effective under specific conditions, it's the issue of funding. But I strongly suspect that the international community, given the WHO endorsement now, will make exceptions where it doesn't have to be used with ITNs or IRS, for example, in humanitarian situations, where it's the perfect tool to be applied under those conditions. For the rest of it, very unfortunately, it's going to depend on end users to cough up and pay for the products themselves. They do work. Outdoor application is still a little bit uncertain. We wish you, Michelle, I'd love to know exactly where you placed the emanators and how far away the people were seated from the emanators when you did your EM landing catches and so on. But that's for a separate off-site discussion. Thank you, John. I hope that answers part of your question.
No, that's very good. That's very nice response to that question. I'm sure these people have similar questions, so this might satisfy anybody with such a questions. There is another question to you, Prof, from the audience, from somebody, Indra. I don't know how to pronounce the share name. And this is an interesting question, which goes, what control methods can we use to control the vectors of zoonotic malaria? They are biting outdoors in the early part of the night and mostly in forest fringe and farms. So how do we control these vectors? Let's entertain. It's a very nice sort of question that's a little bit tangential to the what we but still relevant.
Yeah. So, hello, Indra. Thank you for that question. It's a tough nut to crack, and it applies to many different situations. Forest goers in Cambodia, or wherever you find people, working in rubber plantations or forests for forest products, similar situations. So, zoonotic malaria outdoor transmission. You sit with the same problem, even confronting anthropophagic species, whether they zoophilic or anthropophagic. If they're outdoor biting, it's a tough nut to crack, and we've seen, hopefully, from my presentation that there are quite a number of different tools, but there's not one of them that really has proven beyond reasonable doubt adequate for WHO endorsement that it will help at population level for malaria reduction. However, there have been studies in Thailand and in Cambodia, and I'm sure in Malaysia as well, but there are published studies in Thailand and Cambodia that have shown that a combination of protective clothing, of topical repellents, spatial emanators, again, that integrated approach does help for protection of individuals. It may not translate yet to population, but then, the population we're talking about is a few individuals that are exposed to non-human primate zoonotic infections and so on. So in cases like that, the one that comes to mind is trying to convince people that despite the oily residue and the discomfort associated with topical repellents under certain specific situations will help. Then again, you have to overcome that. First of all, the person has to pay for the product themselves, because the national program or Global Fund is not going to provide that free of charge. And secondly, the psychological barrier of this thing is inconvenient. It makes me sweat. It's not pleasant. Not sure if that answers, John.
Well, that's a very good response, Indra. I hope that satisfies your question. But yeah, I'm just beginning to realize that there is one whole aspects of entrepreneuric zoonotic malaria, and I heard from previous APMEN webinar that the monkey malaria is now becoming quite a problem in Asia, and that it's challenging the effort to control malaria in that part of the world. So this is a very relevant question in this context. Now let's move on to a question for Michelle. Michelle, thank you very much for a wonderful talk on the first ever study of spatial emanator trial in PNG. Okay. It seems you and your team have done very well in demonstrating empirically that indoor spatial emanators reduce mosquito exposure by 50%, am I right? And thus can be used as a complementary vector intervention with ITNs in PNG, if that's the line of thinking that that everybody, I think that's what I'm thinking, that everybody also thinks the same. So what is the next step of your SE study? Are there any plans to conduct a randomized control trial to study the effect of SE products on the impact on malaria incidence, prevalence in humans? Do you know?
Thank you, John. Yes, there are further plans for future work to generate more evidence, especially the impact of the emanator against the local Anopheles population here in PNG. So we plan to do experimental heart trials, which are mostly entomological survey, but there is a planned larger trial where we will look at malaria epidemiological outcomes.
Excellent, excellent. Okay, that sounds really good. I think it's just natural that you progress from entomological effect and then you move on to see how it looks like in the epidemiological sense. Okay, so that's great. Now there is another question for you from the audience, from somebody named Banga Muleba. Okay, and the question goes regarding the spatial emanator Guardian. Are you using Guardian, Michelle?
Yes.
Okay, so Guardian, have field evaluations of their material integrity been conducted? I guess what I mean is like if you put like, how long they last in the field, or like, the integrity out in the field, like, what you can test in the lab versus out in the field.
Well, in terms of like material, the emanator itself, and whether or not it wears and tears, we conducted this study for just a little over a year. And at the end of the study, which we recently concluded the study, the devices were still intact and in good conditions. And the only thing that we observed, or in terms of like degradation, there wasn't any degradation, but there was discoloration due to smoke exposure to smoke, as well as like dust cover. But otherwise, they kept their integrity throughout that year period. Yeah, I guess I hope that answers the question.
Okay. Banga, if you want to ask more question, you can probably send an email to you have the link to the APMEN, and you can post your further questions on that. Maybe then Michelle can handle it more in detail. But yes, thank you so much for posting that question, and thank you, Michelle, for answering it for him. Okay, so we shall now move on to the question for our last presenter, Miss Rebecca Vinit. Rebecca, thank you very much for wonderfully presenting the results of your doctoral research project, which we enjoyed very much. I enjoyed it. I'm sure everybody else would enjoy it too. Okay, so it appears that the residual spray had an impact on the biting rates of Anopheles mosquitoes in indoor and outdoor, but for Plasmodium infection rates in the Anopheles, the effect was only seen in indoor but not outdoor. Now, I know that these are preliminary results and that the outcome might be different when the study is fully complete, but for now, what would explain the lack of an effect on the outdoor infection rates, do you think?
So the infection rates is really driven by the density of the mosquitoes. So having that the biting rates were more effective inside the houses were lower, it does also show that the infection rates would be higher, lower. But for the outdoor, the numbers, it didn't do much effect for the mosquitoes outside. But we're still early into the analysis. We haven't really fully covered all of our mosquitoes yet. And the majority of our mosquitoes, especially from the control villages, we have about 20% more to go through that. So I'm also very keen to see how that will turn out. But in terms of what you've asked about the treatment being significantly reduced with the minimum infection rates for indoors only, that is highly likely compared to the outdoor.
Okay. All right. Now, I was just wondering, like, maybe you will have to suppress the mosquito density a little bit more in order to really see some changes in the
Yeah.
And then maybe like you have to have sufficient reduction in vector abundance or vector density before you start seeing any tangible change in transmission. Sorry, so infection rates in the mosquitoes and probably also in humans.
Yeah.
I'm just thinking, but I may be wrong. Yeah. But anyway, thank you so much for responding. There's now final question from the audience from somebody named Punam Sema Pelamuri. Okay. So the question goes that even though human lending collection is a gold standard, it is usually not approved ethically. So what could be the alternative equivalent of the method if you were to do it, if you were to do the same work with something else, what would be an alternative method?
So why the human landing catch is a gold standard, first of all, is we need to know the human biting rates that will determine the entomological inoculation rates. But to substitute that, you can use the CDC light traps, but then you have to do further analysis on your mosquitoes to actually see if the blood meal that was taken was actually human blood meal, and then you can further analyze that to get them mean biting rates. So you can substitute that with the CDC light traps or any other methods, but you'll have to do that added extra step to analyze to get the mean biting rates for really determining the transmission intensity.
Okay. All right. So that concludes our question for this series today. I hope, Punam, that that Becky had responded nicely to your questions. But otherwise, like I've said with the previous question, that you could also send us an email via the APMEN link that you received when participating in this in this webinar. So that's the end of questions series. Prof. Brack, what are we supposed to do next after this?
Okay, sorry. Thank you, John. Thank you to our wonderful audience. If you wouldn't mind, you'll see up on the screen there's some poll questions, just a very few questions. It's going to take you 1 minute just to respond to those questions on the screen. If you don't mind, just push your cursor onto the screen and respond. It helps us to understand what your impression was of the webinar. So we'll leave that up for 2-3 minutes to give you time to respond to that. But may I also use this opportunity just to thank our audience members for your enthusiasm to register and participate in this talk. We hope you found it useful. May I thank our speakers, Michelle, Rebecca, Ned, for wonderful presentations, excellent, very interesting presentations, and John for your moderation. And then always a special thanks to our people in the background that do a very good job in ensuring that the webinar is up and running smoothly. Dr. Pon, Wulan, thank you for your contributions to make sure that the webinar is possible. My impression is that to those audience members that have posed questions, do not fear. We did not get to all of them, but I think Wulan is going to harvest those questions and circulate it to the panelists, and we will respond to your questions. And the fully responded set of questions will be posted on the APMEN website, where you can go back to in a 2-3 days time to find full responses to your questions. So again, to everyone, thank you for your participation in this workshop, in this webinar, rather. John, over to you. Thank you.
Well, thank you very much for everybody participating. I'm repeating what he says. And I think this officially ends, right? Am I right, Prof?
Yes.
Yes. Thank you.
Officially ends our webinar, and we'll see you again in another round of webinar, APMEN webinar. Thank you so much for your participation. Bye, everybody.
Thank you. Bye.